Good afternoon and welcome to today's QAAFI Science Seminar. My name is Bruce Top. I'm a professorial research fellow in QAAFI and I'm based at the Marucci Research Facility where I'm a plant breeder, breeding peach, plum and macadamia, and I lead the National Macadamia Breeding Program. I would like to start today by with an acknowledgement of country. I acknowledge the traditional owners of, I acknowledge the traditional owners and their custodianship of the lands on which we meet. On behalf of the traditional owners, I pay respect to their ancestors and descendants who continue cultural connection to country. We recognise their valuable contributions to Australian and global society. Before I introduce Katie, our speaker today, I'd just like to mention a few housekeeping things. This seminar is scheduled from 12 noon until 1 p.m. And at the conclusion of the seminar, we will hold a question and answer session. And if you have any questions, please, right from the beginning, go ahead and enter them into the Q&A using the Q&A button at the bottom of your screen. Please note, don't use the chat button, use the Q&A button, and we will address those questions at the conclusion of the seminar. Well, without further ado, I'd like to introduce our seminar speaker for today. It's a pleasure for me to introduce Dr. Katie O'Connor. Katie is a plant breeder with the Queensland Department of Agriculture and Fisheries. She's working in the strawberry breeding team and has a focus on genomics analysis. I got to know Katie while she completed her PhD here at Marucci Research Facility, working in our macadamia breeding program from 2016 through to 2019. And in that work, she started the use for us of genomics and component trait analysis. And once she'd finished that, she went on and did a post-doctoral research in strategies for the implementation of genome-based phenotype prediction in horticultural tree crops. Katie's research to date includes 12 peer-reviewed publications, as well as many industry publications and presentations to engage with industry. So, as I mentioned, it's my pleasure to introduce Dr. Katie O'Connor, and with that, I will hand it over to Katie.
Thank you, Bruce. So I too would like to acknowledge the traditional custodians of the land on which we are collectively meeting today. I live and work on Gubbi Gubbi land, and UQ is on Turrbal Yuggera land. I pay my respects to the elders, past, present, and emerging. And I extend that respect to Aboriginal and Torres Strait Islander peoples joining us here today. So my name is Katie O'Connor. My pronouns are she, her, and today I'll be speaking with you about breeding for better horticultural varieties quicker. So I'll just give you some quick, fast facts about horticulture in Queensland. Horticulture is Queensland's second largest primary industry with a value of over $2.8 billion per year. And just as a comparison, Queensland sugar is worth about half that, $1.5 billion per year. Production horticulture is the most labour intensive of all agricultural industries and represents about 50% of overall operating costs. So you might have been hearing recently about the labour shortages due to COVID causing havoc in the horticulture industry. Queensland has quite a few major horticulture breeding programs, including strawberry, mango, macadamia, pineapples, citrus, papaya, custard apple. But you might not realise that breeding in horticulture is very difficult. For one thing, it's very slow. Often the plants take a long time to grow. They have a long juvenile period, So it can take a few years for them to actually produce fruit, which is often the thing that you want to measure. They can be very large plants. They take a lot of room to grow and trial. It's difficult to get replication in trials. They can be highly heterozygous. You often need large numbers of individuals in the early stages of trials, and then you need to assess the individuals over multiple seasons. Here's just a photo that I took during my PhD of us harvesting a macadamia trial. So we have this pinwheel harvester that you push around under each individual tree. It's like a lawnmower, it picks up all the nuts. So we pick up the nuts under each tree multiple times throughout the season. And then at the end of the season, you take a long stick and it's got like a hook on the end, and then you pull out all of the nuts that are still in the canopy. It's a lot of work, and I am indebted to all of the people that helped me with this through my PhD. But genomics offers an opportunity to facilitate breeding by incorporating DNA data into selection strategies, which can potentially reduce the amount of field work like this. So you might recognize the breeder's equation, where we have genetic gain as a function of selection intensity, accuracy, genetic variance, and generation interval. So one of the main ways that genomics can help increase production in horticulture is by decreasing the generation interval, by allowing breeders to identify elite germplasm earlier in the breeding cycle, which can therefore lead to greater genetic gain. So I'm going to be speaking about macadamia and strawberry breeding, which are my two areas of knowledge, and the ways that genomics assisted breeding are being used in these crops. I'll also outline some of the differences between these two breeding programs. So in macadamias, we've got a relatively small, newish breeding program compared with strawberries, which is larger, more germplasm, more established breeding program. I'll first speak about my PhD research. The title of my thesis was Selection Strategies to Improve Yield in Macadamia Using Component Traits and Genomics. And so as Bruce said, this was from 2016 to 2019, and I had 5 advisors at QAAFI: Bruce Top, Ben Hayes, Craig Hardner, Mabashwa Alam, and Robert Henry. So macadamias. I hope that a lot of you have had the chance to taste them and know that they're delicious. Yes, I did get to eat a bunch of them during my PhD, but you might not know that the nuts come from these large subtropical rainforest trees, and they're actually native to southeast Queensland and northeast New South Wales. You might even have a tree in your backyard like I do. So they're diploid, predominantly outcrossing and largely pollinated by bees. They've also got a relatively recent domestication. Recent research that Craig Hardner was involved in found that a lot of the current cultivars likely originated from a single tree. So historical records suggest that in the late 1800s, someone found a wild tree in Gympie, took a bunch of the nuts from a single tree over to Hawaii, and they bred them out there into a breeding program. So records suggest that a lot of the cultivars are half siblings because they come from a single mother tree. So the Australian breeding program started in the mid-1990s, largely based off these Hawaiian cultivars, and it's based at Marucci Research Facility where I work, and involves DAF and QAAFI. I also wanted to mention that there's been a fair amount of work on the remnant populations and the genetic diversity. My honours research also looked at this. So we've got a large amount of genetic diversity in these remnant populations, despite the fact that they're very highly fragmented. And so this actually offers a really unique opportunity for the breeding program to grab some of that genetic diversity, which just doesn't exist for a lot of other existing breeding programs. So I'll just outline the breeding strategy for macadamias. So originally there was quite a limited pool to use as parents, but new germplasm is produced from controlled pollinations or leaving the trees for open pollination. Then the nuts that develop from these pollinations, they germinate into seedlings, and each one is genetically distinct. So these are grown out in unreplicated seedling progeny trials, and these usually have around 2,000 individuals. So these are grown to around the age of 8 or so, and from the ages 5, that's when they start to produce nuts. From the ages 5 to 8, they're assessed for a lot of different traits. So yield, kernel weight, nut weight, tree size. And then at the end of that, BLUP analysis is performed to identify the best individuals to continue over into the next stage of trials, which we call regional variety trials. So we've got regional, different areas that macadamias can be grown. So this is where we determine how well they perform across different environments. It's really important. So these trials are replicated, and we have about 20 genotypes. So that's about a 1% selection intensity, because we start off with around 2,000 seedlings, and then we end up with 20 selections. So these are grown from, they're assessed from ages 4 to 10 or so for a lot of traits, more traits than in the seedling progeny trial. And then again at the end, BLUP analysis is performed, and an economic index is used to select individuals that are predicted to perform well for industry. So recently, 4 new varieties have been released to industry, and we call them the beetles because they've got really creative variety names, J, P, G, and R. So there's some good outputs, but what's important to note is this whole process took around 25 years from crosses through to release. So you can see why Bruce was really interested in looking at how genomics could reduce this breeding cycle length. So my PhD study population, I looked at a subset of the seedling progeny trial, which is the first stage of trials. And I chose around 300 progeny from 32 families. These had been planted in 2001 across 4 sites, 2 near Bundaberg and 2 near Gympie. And I had chosen my individuals, I chose about 4 low yielding and 5 high yielding individuals per family to give a range of phenotypes. So as I mentioned in the seedling progeny trial, they're assessed from the ages 5 to 8 for yield and other traits. So I made use of historic data during my PhD. And then I also collected data on mature trees. So this is when they were about 15 to 19 years old. I had these genotyped by diversity arrays technology, DART, in Canberra, for single nucleotide polymorphism markers, or SNPs. So SNPs are usually biallelic, and they have 1 nucleotide or allele from each parent. And I used around 4,000 of these after quality control. So the first of the genomic seeks that I'll be speaking about is marker-assisted selection for major QTLs. So genome-wide association study is one way to identify markers to use in marker-assisted selection. So GWAS statistically tests each individual marker for a significant association with a trait. And it relies on markers being in linkage disequilibrium with causal polymorphisms or genes. And GWAS and marker-assisted selection typically work well for major QTLs or loci. So genes that explain a large proportion of the variance for that trait. So I'll give you a quick rundown of some of my results for nut weight, whole kernels, and trunk circumference. These are quantile or QQ plots, and they show the expected significance versus the observed significance of each SNP with the trait. So each of these little dots here is a different SNP marker. This is the one-to-one line. So any markers in the top right corner above the one-to-one line are those that are showing higher significance with the trait than expected by chance. So we didn't have a reference genome at the time, so I don't have a pretty Manhattan plot to show you. But you might be thinking, so what? What does all this mean? Well, I'll give you a very basic demonstration of how we could use these results in marker-assisted selection with nut weight as an example. So if we have a look at these three significant SNPs, I looked at the average phenotypes of trees with these three allelic states, AA, AB, and BB. And for all three of these markers, I found that generally trees that had AA, allelic state for these markers, generally had lower nut weights compared with trees that had medium and large nuts for AB and BB allelic states. So this means that if Bruce wanted to select for trees that had medium and large nuts, he would focus on AB and BB genotypes at these markers. Okay, so let's compare traditional breeding with marker-assisted selection. In traditional breeding, you'd start off with your seedlings, grow them out to 4 to 5 years, which is when they start to produce nuts. And then you would assess the nuts over multiple harvests and multiple seasons. And then based on those results, you'd select the best individuals. So trees that have the nut size that you're looking for. In comparison with marker-assisted selection for QTLs of major effect, you take the individuals, the seedlings, and you just grow them out in the nursery for a year until they produce their very first leaves. And then you can sequence the DNA and then look at the allelic state at those specific DNA locations and then select the plants that have the alleles that you're looking for. So it's much, much quicker. So what about traits that aren't controlled by just a few genes? Well, that's where we can use genomic selection. So this is where genome-wide markers are analysed simultaneously. So I use the genomic BLUP model, which assumes a normal distribution of SNP effects. So we've got negative, 0, and positive effect on the trait. And it looks at patterns in their DNA to predict each individual's genomic estimated breeding values for that trait. So as I said, genomic selection works well for complex traits that might be controlled by many genes of small effect. So we wanted to see how accurately we could predict nut yield in our macadamia population. So I made full use of the historic data for yield from ages 5 to 8. And I used a 5-fold cross-validation with an 80-20 split. So what that means is I took 80% of my trees and I used their genotypes and phenotypes to construct a predictive model. And then I applied that model to the remaining 20% of trees and predicted their phenotype using only their genotypes. And I did that 5 times. So I correlated the predicted phenotypes for yield against their known phenotypes. And the way that we estimate how accurate that prediction is that correlation divided by the square root of the heritability. So I found that narrow sense heritability for yield was 0.3 and our average prediction accuracy was 0.57. And we were pretty chuffed with these results, considering that yield is a pretty tricky trait and we had a relatively small training population. So we can use this information in the breeder's equation to estimate genetic gain. So I compared traditional breeding against genomic selection. So selection intensity. I mentioned that typically it's around 1% because we start off with 2,000 seedlings and we select about 20 of those to go into the next stage of trials. And we also looked at 2.5% as a backup. Accuracy for traditional breeding is the square root of the heritability. Square root of 0.3 is 0.55. And then for genomic selection, the accuracy is 0.57. So we notice that these two numbers are quite similar. Generation interval for traditional breeding is at least 8 years. So as I said, we grow out the trees and assess them to at least age 8. And that's when we decide if a tree is good or not, and if we want to use it in cross-pollinations. In genomic selection, generation interval is 4 years. So yes, we might be able to predict which individuals may perform well before that, but we still need to wait until the trees are reproductively mature, so flowering, so that we can use them as parents. So we plug this into the equation, and then we had an estimate of genetic gain in grams per year for traditional breeding and genomic selection. And you can see that genetic gain for genomic selection is about double that of traditional breeding. And this is largely due to the fact that generation interval is halved. We've just had a paper published in BMC Genomics just 2 weeks ago, if you'd like to check that out. So I'll compare the two breeding methods again. So you start off with individuals in the seedling stage, grow them out for at least 8 years until they're mature trees, and you're assessing yield over multiple harvests and seasons. And remember, that's when there's a lot of work involved in the field. And then based on those results, you select the best ones. But in genomic selection, similar to marker-assisted selection with major QTLs, you grow them out in the nursery for a year or so, and then you sequence their DNA, and then you apply the predictive model to those individuals to predict their genomic estimated breeding values. So this is when you can either select the best individuals to carry on testing or to use them as parents, or you can discard the individuals that are predicted to perform badly. So that means that you're not wasting land and labour on material that you don't think will perform very well. So it's years quicker. It's really promising. But I need to mention that we still need the second stage of trials, the regional variety trials, to determine how well they perform across the different environments. So the next steps. Firstly, these results need to be validated in a separate population before putting them into practice. But in terms of putting them into practice, we understand that genotyping is quite expensive, and at the moment, it's not feasible for us to genotype all 2,000 of the seedlings in the seedling progeny trials. So Bruce has a game plan to focus on a subset of individuals. So first off, you identify which of them are precocious. So you grow them out until they start flowering and producing nuts. So this is a really important trait because you can imagine growers, they've planted the plants and then they just wait around for years until they start producing nuts, and that's when they start making money. So trees that produce nuts earlier on means quicker returns on their investments. So the trees that are producing nuts early on, you can assess those nuts and determine which ones have high kernel recovery. Kernel recovery is the ratio of kernel weight to nut and shell weight. And this is another really important trait because high kernel recovery, around 40%, earns a higher price per kilogram for the growers than low kernel recoveries, which is around 30%. So using this truncated selection, where you've got precocious individuals and those with high kernel recovery, you get a subset of the individuals. And you could genotype those and then apply the predictive model for yield to those individuals. I believe this is quite a common approach in tree breeding due to high genotyping costs. And then this could also be applied to more traits like trunk circumference. So I measured trunk circumference on mature trees as an indicator of tree size. So you can imagine that smaller trees would be beneficial because you can plant them closer together and have higher yields per hectare. You can see it's rising off the 1-to-1 line here, which didn't happen in these two traits. And we believe this is an indicator of polygenic inheritance. So I found that there were 44 significant SNPs, but multiple regression analysis suggested 16 separate QTLs. So we think that there's quite a few genes involved in this. which makes sense. So this could be a good trait to try with genomic selection. Firstly, I wanted to say a huge thank you again to all of my advisors for their support during my PhD and afterwards during publishing. The macadamia breeding program is funded by Hort Innovation, and I had a lot of people helping me during my PhD. This is me on my last day of fieldwork for my PhD, and I'm very happy and I'm outstanding in my field. Anyway, now onto my current work in the Australian Strawberry Breeding Program, which is led by Dr. Jodie Neal. So strawberries are also delicious. I've got a pretty sweet gig. I'm very lucky in my jobs. Strawberries are mostly outcrossing as well. They're mostly pollinated by bees, and they're octoploid, but we're lucky that they effectively act as diploid. They're usually an annual crop, but some growers let them return for a second year. And unlike macadamias, they have a fairly long breeding history. So the Australian Strawberry Breeding Program, we're a national breeding program. We breed for 3 different regions across Australia. We've got subtropical in southeast Queensland and Bundaberg, temperate around Stanthorpe and the southern states, and then Mediterranean near Perth. We aim to produce robust, attractive, flavourful varieties that are profitable for the growers. So that's a really important factor. The growers need to be able to make money to produce food for us to eat. And our varieties have been performing really well recently. In 2019, they made up about 90% of all strawberry plants grown in Queensland. And that had a farm gate value of about $150 million. We are also the biggest breeding program funded by Hort Innovation. There are quite a few different strawberry breeding programs across the United States and Europe. So you might have heard of Rosebreed, which is a consortium of research groups studying rosaceae crops, which strawberry is part. The University of Florida have a really big program, and we actually have a seed exchange program with them to keep up our diversity levels in our programs. There's an Axiom SNP chip array. There's 2, there's 90K and 35K. So there's quite a lot more resources available in strawberries compared to macadamias, because it's a big crop and there's lots of people working on it around the world. So I'll just outline the strawberry breeding program and the strategy. So we first start off by talking with, I should say, listening to growers and industry to define the target traits for each region. And the target traits are slightly different between the 3 different regions. We have quite a detailed process that we use to select parents to cross. So there's controlled pollinations, and then each seed on a fruit is genetically distinct. And we literally put fruit in a blender with a bunch of water and then sieve out the seeds and then germinate those. So the seedlings are planted out in the field. In the subtropical region, we have about 15,000 seedlings, and same for temperate. These are unreplicated, and we don't collect any data on these. Every month through the season, we just do a walkthrough and put a pin tag in any plants that look like they're performing well for a bunch of different traits, just based on how they look. And then we select the best 150 or so at the end of the season. So similar to macadamias, that's about a 1% selection intensity, going from 15,000 to 150. So these are clonally propagated by runners and planted out in replicated clonal trials. And this is when we assess the traits, weekly and monthly. So every week, the ripe fruit is harvested, and then we get values for yield and average fruit weight. And then throughout the season, every month, they're assessed for a whole bunch of traits like internal colour, external colour, flavour, bricks, acid, resistance to rain damage, a whole bunch of things. Then at the end of the season, we do a big BLUP analysis and we get an estimated economic value for each individual. We select the best lines based on these results, about 30 of them, to put into the second stage of clonal trials. And then we do all of that again a second year on those 30 individuals. Then at the end of the second year, we select the best individuals to go out into on-farm trials. So we take around 2 to 10 of those and we give them to the growers for them to trial. So this is incredibly important for us because we need to know if the growers like the varieties that we're providing to them. So they give us information on yield, quality, pack out percentage, things like that. And then based on their feedback, we'll decide if we release any of those. So this whole process takes about 5 years. We've got seedlings, first stage clones, advanced stage clones, and then 2 years of on-farm trials. So it's quite a bit quicker than macadamias. Also, these 5 stages are running concurrently every year for the 3 regions. So we are busy bees all year round. We've been using genomics assisted breeding techniques for a few years in our breeding program. For micro-assisted selection for major QTLs, other research groups have developed markers, SSRs, simple sequence repeats, or microsatellites, not SNPs, for these traits. We've got the peach flavour volatile and the sherry flavour volatile. So fruits that have these 2 volatiles have a superior flavour than those that don't. We actually found that the sherry flavour wasn't stable for us, so we've stopped using it. But we also use this process for bearing type. So short day plants versus day neutral plants. And what I mean by that is short day plants have a really big burst and they produce a bunch of fruit in a short window, whereas day neutral plants have a trickle of fruit throughout the season. So you can see how it's super important for the growers to know which variety they're dealing with in these 2 bearing types. These traits are also quite tricky to phenotype compared to, say, average fruit weight, and you need specialist equipment like GCMS to measure the flavour volatiles. So it's really useful having a marker as an indicator of phenotype. So I'll just give you an idea of how this works for SSRs. This is a gel electrophoresis, and we have presence of the marker here for this individual, but it's absent for this individual. So it's quite a bit different to looking at nucleotides of SNPs. And we use this presence-absence information as an indicator of phenotype in our selection decisions for all of our advanced clones, so the second stage of clonal trials. I'll just give you a comparison with the University of Florida, who we have a seed exchange program with. From what I understand, they have a much larger budget than us, and they perform marker-assisted seedling selection on 60,000 seedlings every year. So apparently they've got this really cool streamlined process where they set up all of their seedlings in 8 by 12 trays, like a PCR plate, and they just push them through a micro-assisted selection for a bunch of different traits. It's very cool, but it will cost quite a bit of money. Okay, so micro-assisted selection is also expanding into SNPs for strawberries. So this is a photo of a plant affected by charcoal rot, which is a problem in Australia and in other countries around the world. Another research group recently found SNPs associated with resistance for this trait, and we are now in the process of determining if their findings can be applied to our germplasm. This has also been done by East Malling in the UK for powdery mildew. Powdery mildew is an issue in strawberries and quite a few different crops. I'll be doing a genome-wide association study soon to see if I can find any SNPs linked with resistance in our germplasm, and then I can compare my findings with those of East Malling. And then from that, we can look at developing primers so that we can screen for resistance in the lab rather than having long and expensive experiments in the field or in the shed. Okay, genomic selection. Craig Hardner and Malusu from QAAFI have developed a multivariate model for us. So there's 11 traits. We've got season yield, bricks, average fruit weight, acidity, all in one big model. So if you look at these orange columns, this is the predictive ability for the multi-trait model. And you can see that it's all quite a bit higher than for the green columns, which is predictive ability using the single trait model. So this shows that using information from other traits improves predictive ability in the multi-trait model, even for traits where yield is low. And I've recently put this genomic selection into practice for genomics assisted parent selection. So our 150 seedling selections from last year, I genotyped a plate of them, and I applied this multivariate model to those individuals to predict their breeding values for all of these traits. And then I've selected 6 of them to use as parents this year. So these are plants that haven't had any data collected on them at all. And so we're using them as parents. So that's a whole year earlier than we'd be able to do without genomics. So we'll just have to wait and see how well selections themselves perform this year, as well as the performance of their offspring next year. Now for some exciting things. So we've got in the pipeline several varieties for alternative fruit colours and flavours for market diversification. So this is our blush variety that's going through PBR at the moment. It's so beautiful. You might be looking at it thinking that it's not ripe, but it is. So it's pure white inside. It's so sweet. It's not very juicy, which means that it has really good bruise resistance and resistance to rain damage. And this peach colour on the outside is produced from where the sun hits it. So there's this really cool photo that shows an outline of calyx leaves from where the sun has hit the rest of the fruit. They're so beautiful and they taste amazing. So keep an eye out for them in the next few seasons. So we have a very large, hardworking team across the different production regions. I'm fairly new to this team and they're all so wonderful and so great to work with. So thank you to all of them. Thank you to Craig and QAAFI for this opportunity, and thank you to Bruce, not only for introducing me today, but for also being a really supportive principal advisor through my PhD. Thanks.
Yeah, thanks very much, Katie, for that presentation. We have time now for some questions that have been coming through on the Q&A button that's at the bottom of your screen. And if anyone else would like to add some questions, it's not too late to do so. So we have a few here. I'll just start off by asking Katie, let's see here. There's a question here regarding the strawberry breeding targets. At what stage of new accession development is consumer assessment done? Or is it assumed that sweetness, flavour, etc., as previously found by historical consumer information, is to be used?
Okay, I'll answer as best I can. So consumer assessment is done throughout the season on the clonal trials. So that's done by me and Jody tasting the fruit and seeing what we like about them. And I understand that's from a breeder's perspective, but it's also diversity in what we like. So for example, Jody might like slightly tart fruit and I love anything sweet. But then we also work with consumer panelists where they do they do projects looking at what consumers really like and whether they like colours of darker fruit or lighter fruit. And that's in our later stage, the varieties that are given to the on-farm trials. So some feedback that we got from that was quite interesting. The really dark fruit that I showed you in my last slide, the main word that was used with that was sexy, which is not something we're going for, but it's interesting to find out what the consumers like.
That's great. Thanks, Katie. Another question here is, how did you come up with the variety names for macadamia?
Oh, I didn't. But I think, Bruce, it was just A to P, right?
Yeah, that's right.
Yeah.
Yeah.
So it just happened to be the Beatles initials that were chosen.
Yeah, good. So there's another question here. With precocious flowering, isn't that more related to the length of their juvenile period? While for growers, it is less relevant because they will be planting vegetatively propagated adult material.
Great point. I think that there is still a gap from planting cuttings from adult material to them producing nuts. I don't think it's as long as seedlings. And there's a trial happening at the moment at Marucha Research Facility about juvenility and length to producing fruit from adult material. Yep,
and that's exactly right. And so we're really interested. We've got the trial set up now to have a look at if seedlings that have a short or a long juvenile period, how does that correspond to their precocity using adult grafted material from the same genotypes. So hopefully that'll answer that question. Yeah, there's a question here regarding the markers that you've used, that you mentioned that you used dark markers for macadamia, but what type of markers did you use for strawberries?
Okay, so we use the Thermo Fisher Axiom array through Genotyping Australia in WA.
Yep. Another question, is the antioxidant level lower in the paler coloured strawberries? I'm asking this as some consumers have been trained to see high red colour as being closely associated with higher antioxidant levels.
I think that might be the case, but there's still research happening about that. And I think, I hope I get this right, there's a lot of different antioxidants and some of them are not associated with the colour, in that pale coloured strawberry, so that beautiful blush one, actually has a really high amount of antioxidants. I can't remember which one it is. But there's a group of researchers at DAF and then another one at QAAFI that are working on that.
Okay. Another question. Do you think that genomic selection can be a substitute for CRISPR-Cas?
A substitute?
Mm.
I don't know a lot about CRISPR. I think that it offers some really great opportunities to speed up the breeding program. So say you have a trait that you want to breed for and you have it in some material, you could take your time to back cross, integress, try and get that gene, that trait into your germplasm, and that would just take ages. But with CRISPR, you might be able to do it really quickly. I don't really know what's involved in that. I don't know if you'd say it's an alternative. I think it could be used with it. That's out of my area, I'm sorry.
No, and there's a follow-up question, which is your team considering using genome editing technology to speed things up even more in the future?
I definitely want to look into that. Not at the moment, and I don't know if those sorts of technologies have been used in strawberries in other breeding programs. I'm not sure, but I'm interested to look into it.
Do you know if it's been discussed with the strawberry industry, Katie?
I don't know, and I think it would depend on who you're talking to and the level of understanding about genome editing.
Yep. Good. There's quite a few comments saying what a wonderful presentation. And
oh, thank you.
There's an interesting one here. You've been working in this topic and published quite a few papers, but if someday you decide to leave academia, is there a demand in industry for researchers with knowledge in genomic sequencing, genotyping, and marker detection?
Okay, so I think that working for the government, which I do now, is outside of academia and outside of industry. It's sort of in between. I like working for the government because I'm still involved in the research for genomic sequencing and things like that, but it means that I get to directly apply that knowledge to the outputs that are required as part of working for the government. So I think I'm in a good place to keep doing what I am doing.
Yep. Another question related to macadamia. Is there an attempt for a reference genome assembly?
Yes, there is, and there's two versions of it. I hope I get the details right. I think they've got them down to 4,000 scaffolds, which is pretty good for 28 chromosomes. Is that right, Bruce?
And there's more work happening with in Rob Henry's group with the sequencing that's been happening. Priyanka's doing a comparative genomics study where she's sequencing representatives from all 4 species of macadamia, and so that's really really improving the reference genome for all the different species.
Yeah, so there's 4 species in the genus. 2 of them produce pretty inedible nuts. They're not used in breeding very much. And then the other 2, Integrifolia and Tetrafolia, are used in breeding quite extensively, and they're hybrids.
Yep. I think that's about it, Katie. Just looking through here. There's another question here regarding the value of pedigree-based predictions versus genomic-based predictions, and whether you've considered, one versus the. What's your thoughts on the comparison of one versus the other?
Yeah, so until recently, pedigree-based predictions have been the pretty standard way to go. So using information about pedigree to more accurately predict the performance of individuals with BLUP analysis. So it works well, but if you've got the funds to do some genotyping, analyses using genomic information can be more specific, more accurate, because they're accounting for Mendelian inconsistencies in relationships between individuals. So for example, full sibs or parent offsprings, they're related at 0.5. So you'd be basing your pedigree-based analyses on 0.5 relatedness. But in genomics, you can actually estimate what is their relationship between those. And it might be 0.46 to 0.53. And then you've got like the individual SNPs that are similar between individuals. And it's much more accurate predictions.
Yep. I better finish up here, so we make sure we finish on time. Oh, hang on, there's another question just coming in. Are all the traits of interest simply controlled genetically, and so accessible via these techniques, or do we need to introduce more biological insight?
Oh, absolutely. We definitely need to look at the biological processes involved in this, and my knowledge only goes to a certain point. So I can look at traits that are controlled by a few genes, so micro-assisted selection with major QTLs, or traits that are more complexly inherited, so like yield with genomic selection. But then I don't have the knowledge of how to look at what are the genes and how do the genes work and what are the processes involved in that. So I think it would be really beneficial to link up with teams that can help me take the next step and then the next step and then the next step. So yes, some more biological insight would be hugely helpful.
Good. Okay, thanks again, Katie, for your presentation. I'd just like to advise everybody that next week's seminar is on Tuesday, the 8th of June, and it's titled Juicy, Succulent and Tasteful Pork Made in Australia, and will be presented by Marta Navarro from QAAFI's Centre for Nutrition and Food Sciences. And so just visit the QAAFI Science Seminars webpage for more information on that coming seminar. And I'd like to close up now and thank everybody for attending today. And if you enjoyed the seminar, please use the poll at the end on this end slide, and sign up if you haven't already for the rest of the seminar series. So thank you, everyone, and thank you, Katie. And
thank you.
Goodbye.